Hello, welcome. I'm Olivier Paquatte, Head of Scientific and Technical Development for Personal Care at Safic-Alcan. Thank you for tuning in. Today, we are diving into an exciting question at the crossroad between biology and cosmetics. How can biology shape up the future of cosmetic science? There's one timeless question we all ask ourselves. How can we live longer and age better? Over the past decade, brilliant scientists around the world have made incredible breakthroughs by exploring the biological mechanisms behind aging. What if we could capitalize on that knowledge and apply it to skincare? In this podcast, I'll guide you through some of the most fascinating biological pathways that are already reshaping how we think about cosmetics ingredients. From DNA repair and telomere extension to microbiome, exosomes, neurobiology, and even cellular reprogramming. You'll discover how cutting-edge science is unlocking new ways to support healthier and younger-looking skin. Stay with me because some of the insight you'll hear might just change how you think about aging and how we can fight against it. Let's get started. Addressing genomic instability is an illustration on how biology can help cosmetic science. Scientists are convinced that aging is a loss of information. DNA function is to store and transmit all the biological information necessary for the body to function. Research efforts are invested in identifying which are the key factors that can damage DNA and how to fight against these factors and how to repair potential damages. For instance, hyperreactive oxygen species have recently been identified. They are generated by UV, blue, red, or visible light, pollution, stress. Artificial intelligence is being used to identify the most effective molecules able to fight against hyperreactive reactant species. AI also helps identify synergies between thousands of natural extracts and tens of existing vitamins and peptides. Also, focus is given on elements such as sirtuins, which have been shown recently to be critical to control the epigenome and DNA repair. The latest research focuses on identifying the smart ingredients which can locate and repair DNA damages associated with aging. Marc-André Sellos from the Muséum National d'Histoire Naturelle said in a recent congress in Paris that death is today the unavoidable endpoint for our cells, and therefore for us. Immortality is granted for only one part of us, our genes. We have hard evidence that short telomeres can cause senescence. By the way, do you know that there are methods available that can measure the length of your own telomeres? A very short telomere will lose its histone packing. As a result, DNA at the end of the chromosome become exposed. This leads to major hypergenome instability. Recently, some sirtuins have been shown to help extend telomeres and package them up so that they are protected from degradation. Today, AI-driven screening programs are in place to identify active ingredients specifically targeting these sirtuins or able to extend the length of telomeres. Epigenetics sounds like science fiction. This is not. It corresponds to the modification of gene expression by outside factors such as pollution, stress, UV, sleep, food, physical activities. This does not involve any mutation. In its native state, DNA is wrapped around histone proteins. It is compacted into chromatins and folded further to form chromosomes. Chemical modification catalyzed by enzymes such as histone methyltransferase results in the modification of histone proteins. This can control whether chromatin is tightly packed and silent or loosely packed because of the chemical modification and therefore actively expressed. C. elegans is a small worm made of about 1,000 cells. Histone methylation due to the presence of a specific enzyme leads to 30% longer lifespan versus the wild type. In bees, the differentiation between the worker and the queen is a fine example of epigenetics. During the post-embryonic period, the queen is exclusively fed on royal jelly, unlike the bees, which will become workers. The inactivation of specific genes turns selective bees into fertile queens. In 2010, gene sequencing results showed that between the queen and the workers, 560 genes are differently methylated. This results in very different characteristics for the queen, like much higher weight and much longer lifespan. These are two examples where biological pathways provide benefits, such as longer lifespan. Interestingly, clinical studies demonstrating that epigenetics has beneficial effect on skin health have recently been published. Senescent cells are often referred to as zombie cells because even though they should be dead, they refuse to die. Small numbers of senescent cells can cause widespread damage. Even though they stop dividing, they continue to release inflammatory cytokines that attract immune cells such as macrophages. Senolytic ingredients specifically designed to target zombie cells have been shown to slow down aging. We can imagine teaching our own immune system how to target senescent cells. Recently, it has been shown that human immune system has the potential to prevent the accumulation of senescent cells during aging to achieve longevity. This opens doors to senescent cells clearance by boosting immunity to combat skin aging. How about designing a vaccine against aging? There are several hundred of receptors in the skin. We are still trying to understand the role played by more than half of these receptors. Amongst them, olfactory receptors, similar to those found in the nose, have been identified in the skin. Through the modulation of these olfactory receptors, we've been able to limit cellular senescence and improve longevity mechanisms in skin cells. There are millions of microorganisms in and at the surface of our body. In the guts, we have recently identified psychobiotics, which are bacteria able to produce feel-good hormones. One bacteria has been shown to induce senescence. Also, we know that bacteria that are able to exchange genetic material in a way that good bacteria may become bad bacteria in the presence of detrimental bacteria, and vice versa. Skin microbiota itself plays a key role in the skin aging process by enhancing hydration, reducing wrinkle, fortifying the skin barrier, and globally reducing age-related skin symptoms. Dendritic cells, which are part of the immune system, have been shown to target harmful skin microorganisms while supporting beneficial ones. The dermal immune system is able to balance good and pathogenic bacteria to avoid excessive inflammation. Work is being done to identify and fight specifically against those bacteria linked to senescence. No doubt we'll be able to soon identify bacteria that are able to protect DNA, to stop zombie cells and stimulate stem cells growth. Exosomes are very sophisticated vesicles used by bacteria, yeast, algae, and even human cells. Their role is to boost cell-to-cell communication and defense. Exosomes issued from plant cell cultures have been shown to contain a high concentration of bioactive peptides, which exhibit potent anti-wrinkle effect and regenerative properties in skin and hair care. Contrary to the brick-and-mortar models, keratinocytes are not just building blocks. Recently, it has been shown that they are synaptic-like structure at the surface of keratinocytes. Also, we have seen nerve fibers going across these epidermal skin cells. Recent data have shown that activating nerve fibers can boost collagen synthesis. With aging, the density of nerve fibers at the surface of the skin is decreasing. This has a negative impact on collagen synthesis. It promotes aging. Work is being done in identifying ingredients able to keep the nerve fiber density at the surface of the skin as high as possible. This illustrates that neurobiology offers some interesting leads in fighting against aging. What if we could reset the aging clock and prevent cells from losing their identity and becoming senescent? Indeed, the solution to aging could be cellular reprogramming. In 2012, Dr. Yamanaka won the Nobel Prize in Medicine for identifying a set of 4 genes which could induce adult cells to go backward in their development process and become pluripotent stem cells. Research is being done to see whether we could use these and other switches to reset the entire body epigenetics landscape. Cells that have lost their identity during aging can be led back to their true selves via cellular reprogramming. We can imagine that gray hair would disappear, wounds would heal faster, wrinkles would fade, the whole body would feel young again. Some specific plant extracts have been recently shown to activate some skin stem cell reprogramming pathways. We are still at the infancy of exploration of this specific biological pathway. I hope you enjoy this glimpse into how biology is putting cosmetic science upside down. Thank you for listening. And don't hesitate to visit us at the Safic-Alcan booth to continue this conversation. We also look forward to welcoming you throughout the year at our various events and customer days. Let's keep shaping the future of cosmetics together.